Hello everybody, this is Justin Borbitz, the very last lecture for your second semester of lockdown. You've survived, congratulations. Biol3157 Bioinformatics, just taking you away here with some closing thoughts, recording from the beach down in South Thuris. Hopefully this is a way to communicate without interruptions and a bad signal. So let's jump right in. What have we learned so far? Look, the deluge of genomics data is overwhelming. We have, to start a whole new course on teaching you how to deal with this data. A lot of people are resistant to jumping into genomics because even though they might be able to afford it and generate the data, they often don't know how to analyze it. And so bioinformatics is now the rate-limiting step, and what we need you to know is not just arrive on the scene with a huge amount of data and have a go at it. You really need to be involved in the design and think about how the samples were selected to be sequenced, what the technology was. And so then we can proceed with hypothesis testing and actually make some sense out of the data. So, this is the main point of bioinformatics is to analyze genetic sequence data. You want to know about that diversity matrix, samples and variants. Usually we have hopefully thousands of samples, but usually millions of variants, sometimes even hundreds of millions of variants from gigabases of DNA. So where do these samples come from? That's a starting question, that the samples are going to be related to each other. You'd like to figure that out. You'd like to know where they come from, understand isolation by distance, and then get at the more important, potentially adaptive piece, which is how the environment filters genetic diversity. There'll be some limits. You can have overall genome-wide isolation by environment, and that'll affect all the genes in the genome. Same way that migration is limited by gene flow and geography. We would like, though, to go beyond and get at the adaptive alleles, and that's what whole genome data can do, separate the migration from adaptation. And so it used to be that we started with collections on the shelf that we didn't know much about. We would measure them, and the very last, most difficult piece was to sequence them. But now that's really the first step, and so we go through our collections to find the most important core populations that are worth investigating phenotypically, growing in a controlled environment if you can, so that the phenotypes that you measure are due to the genetic basis, the underlying genetic traits, rather than environmental variation. When you just measure plants or animals or people in the wild, they have a lot of phenotypic plasticity that's hiding. The environmental variation is, in that case, noise. It's hiding the true genetic diversity. One way to get at that is longitudinal studies, multiple developmental stages. COVID, we thinking a lot about. It's really an elderly disease with pre-existing conditions, so you need to look at that phenotype over time and understand how the genetic causes are conditional on development as well as environment. And of course we're talking about whole genomes, and so we need to always keep in mind that there's a collection of variants that are neutral, and they're correlated with each other because of non-random mating, this population structure. It's not always a perfectly balanced shuffled deck. We've got some biases in every real population, and we need to account for that. And once we do, then we can get at the causal variants, the adaptive alleles, which are only a few percent of the variants in the whole genome. It could be thousands of important variants out of millions of potential variants. So we are looking for needles in a haystack, and if we don't control for the background variation, even when it's small, we'll still get a lot of false positives. Okay, so again, samples and SNPs, this is how the raw data comes in. We certainly like to know where the individuals come from and any other information, and we'll get to that. But the first level of analysis is to do that correlation, samples by samples. So this might be 1,000 by 1,000 or a matrix of 1,000,000 pairs of samples. And what we find in there is these closely related individuals that are families, and very distant related individuals, a deeper population structure, like Africans versus Europeans, and so not the subspecies, but deeper population structure, ancestral variation. That's really important for conservation, and we'd like to think about that. We also we want to, in seed banks, preserve some of these ancestral individuals and try and keep them alive and breed those valuable resources. And that's really the goal, is then to filter down the initial collection of individuals to a core, what could be considered random or closer to random mating population. So then we can start to look at the SNPs themselves. So that's an important topic. We're not going to move on to analyze the full set of samples. We're probably going to do a little filtering to collapse, choose one individual in each family, and maybe split off some of these divergent groups that are underrepresented so that they're not confounding the study. And when you have a nice, ideal, well-balanced population, then we can turn the decks, start looking at the SNPs. Now, to do all pairs of variants correlated with each other, this is almost untenable. If it's millions of variants, then 1,000,000 by 1,000,000 is a matrix of 1,000,000,000, and that'll break most computers still, even today. Certainly, if you have hundreds of millions by hundreds of millions, then you're up in the quadrillions of pairs. So we won't do all possible pairs, but you can do a sliding window. You can look for just SNPs on part of a chromosome, say, top of a chromosome, and then moving down the window and do regional correlations to look for patterns of genetic variation moving down the chromosomes. And I showed you videos about those blocks of linkage disequilibrium and brachypodium. And the large blocks are probably non-random. There will always be some large and some small, but they should sort of fit a normal distribution in a neutral model. But if there is a gene that's important, it could be on a large haplotype block, a large segment that's just swept through the population due to strong selection, positive selection. The other thing you can find is almost a very low linkage disequilibrium, unusually small blocks or absence of blocks, and that might be due to extra recombination, but that's known as balancing selection, where there's multiple beneficial alleles and the heterozygous advantage. But mostly when you're scanning the genome, we'll see that background recombination rate, the normal size, the well-shuffled deck, how shuffled is it? If it turns out that there's recent history in your sample set and not a lot of outcrossing, then you could have large blocks of linkage disequilibrium, which is great for detecting significant associations. We're not killing ourselves with millions of independent tests. These blocks reduce it, say, to 10,000 independent blocks in a genome. Humans have large LD blocks. We've, recently, in evolutionary terms, kind of risen to large populations, and so there is extensive LD. That's good for detection, but bad for identifying the actual causal variant. And so people are going back into ancestral populations then where there's more recombination on average to try to get down to the causal locus. Okay, we wrote about this years ago, the missing heritabilities in the field, Ben Bracci et al. This is a pretty highly cited paper now, which is theoretical. Step A there shows you the kind of conceptual populations, there's layers of structure, and then B is a approach to what I call restructuring the population by just collapsing a lot of these recently founded groups. You might need to shuffle up those lineages. If there's a lot of population structure there, you can make crosses between pairs or intercrosses between these ancestral lineages to reshuffle the deck. Alternatively, you can focus on specific subsets. This is known as regional mapping. You can just split up the group and resample. Go back to the field and these hybrid zones, fill in more populations. Certain ones will be more informative than others. And this is why we really take a step back. Don't just start your study with whoever walks into the hospital or whatever you have on the shelf. Go really back to the wild and try to understand the ancestral genetic diversity. Then focus on the samples that are most informative to study. Here's the example from Brachypodium. We had a few hundred samples that we looked at whole genomes. They were easily collapsed into some clone groups, close family members, like identical, even though their names were different. Sometimes even from different fields, the same plant would be found because it does spread that way. And then there's a few outlier strains, which we only had 3 of. It would be nice to find more. That's another study for another day, but we needed to set them aside for this. And we still had 2 groups to deal with. So we could collapse it down to a working population that we had to deal with the deeper structure statistically, whereas we had independent individuals on the tips. So this is how you make a core population. If you turn the deck now and look at the SNP associations with each other, you can find these correlations. This is a SNP by SNP table focused on a central one, and we just moved down the window in say 5,000 steps. So that's an LD block in yellow. If you want to look at the R squared is the color, so a high correlation coefficient, R squared, means there is a non-random association between those SNPs. That usually happens when they're next to each other and there hasn't been time for recombination to break it up. If you plot that R squared as a function of the distance, the physical distance between the SNPs on the chromosome, you can see in red how that distance decays. And you want to know the distance half maximal. That's the decay with distance in red. So it decays from kind of an average of 50 on about 25 kb for that top of chromosome 1. And over on, this is just the middle of chromosome 1. If you look at a different part of the chromosome, the LD decays to half maximal at greater than 200 kilobases. So that's a really big LD block. So this kind of moves around the window and you can evaluate it in steps. You use a lot of data to fit that LD decay in the windows and move down the genome. So that's how I identify these selective sweeps, potentially in the middle of chromosome 1 and a normal background LD size in chromosome 2. All right, so that's just population genetics from the first week, just to review how we learn about populations and then how we learn about parts of the chromosome. So let's bring in some more information now, because if we measured these individuals for their phenotypic traits, or we know where they come from, then we can start to get at the functional variation. We can also look at other features about migration, isolation by distance and isolation by environment, so that they, at what range do populations start to differentiate? What is the distance of gene flow? So let's start with the GPS coordinates, where the primary collections come from. Sometimes somebody collected it in the field, hopefully in a more or less natural condition in the first place, and you have that information. Most stock centers have some unknowns and some falsely identified. I got it from you, you got it from me, we sent it around the world. And So we can't expect every location to be correct, but as long as most of them are, I think we can exclude some outliers from this study as well. The genotype data is still an interesting individual, but we don't want to trust the information where it comes from. This is on the right, our eucalyptus collection so far for the most recent study. We got over 1,000 new collections, and this is a real resource in Australia to go across clusters of species and build up our natural seed banks. So we're in the process of going through this for genetic diversity now. So the idea of making collections and analyzing their genetic diversity is to build populations that are both adaptable, have background genetic diversity, and are adapted to known environmental conditions that hopefully are relevant for the future. Maybe we can make some climate-ready crops or hardier strains. There might be myrtle rust, for example, is another grant that's taking down a lot of eucalypts and other species in the Myrtaceae. The paper barks are sensitive and dying from myrtle rust, and there is resistance out there, but we'd like to identify the causal genetic basis for that. If we could have a marker on a resistance gene, that's really helpful for the botanic gardens that are trying to amplify resistant seed stocks. But we don't want to take one winter and replant New South Wales coastal ranges with seed from a single tree. Even if it was truly resistant, we made no mistakes, there would be other pests and environmental extremes that would knock back that monoculture. So we need to be both adaptable, have the background diversity for who knows what, as well as adapted to the most important genes that we know about and conditions that we know about. So it's breeding and selection for things that we know about, and balancing, buffering, capturing and conserving the background genetic diversity, which probably has some function somewhere someday. All right, and to do both of these, it is called adaptive introgression. We'd like to move these resistance genes into diverse backgrounds. Sometimes nature's done that already, and we can just sort of help select those lines and enrich their gene frequencies in the next generation. So now for the GWAS part, the associations with phenotypic traits that you measure. When genes are causing phenotypic variation, they're heritable traits. It's not going to be 100% heritability, and sometimes flowering time, for example, in Arabidopsis can be 90% heritability, so very little environmental variation. But sometimes the traits are only like human body height. Human height is now probably 60% heritability. So those are pretty good traits. Other things like even type 2 diabetes might only have a couple percent heritability. There's still knowledge that we can get in big studies about the underlying genetic basis, but really with low heritability diseases, it's all about managing your lifestyle. So that's the overall heritability. Now we'd like to know, what are the causal genes? That's when we start scanning, and we need to measure our populations. These core mapping populations will be the most informative for studying. Now, in human trials, we don't really get to choose who comes into the hospital, but we'd like to, and certainly in plants, that's a real advantage. And also animal breeding, and even for microbes, you can choose a core diversity set, right? For Brachypodium, we have that now, and that's what goes into the growth chambers for phenotypic analysis. So it comes after the landscape study. We're going to grow these in common conditions to minimize the environmental noise, but we might grow them in contrasting environments to get that G by E, that signal of how the different genes control phenotypes in different environments. So the same gene may not make the same allelic variant. It could be two allelic variants. and they have no difference in one environment but a big difference in another environment. So and that can be also true for developmental stages. This gene controls growth in adolescent phase, and then sort of disappears later in life, and other genes are important in later developmental stages. So we'd like to look over space and time. The development is the time, and space is this kind of macro environmental signal. When we get this done, and I'll go in later on into different slides, when we get the data, when we have genotypes, when we have phenotypes in different environments, we can put this together in a model. And then we have an estimate of how a gene controls a trait. And so if we look at a plant that we've never measured before and all we know is its genetics, we can compare that to the plants that were in the training data, the individuals that were in the study design, and make predictions. That's how it works in human genetics. We're often not part of a clinical trial, but you can still go to the doctor and get a prediction based on your genetics, and that's how you're related to the individuals that were in the study, and therefore we can predict your phenotype. Now, if you've done this in multiple environments, this is also pharmacogenetics. People would say, what will happen if you take this drug? Well, some people are more sensitive and some people aren't, and we'd like to know the genetic basis for that. And so if we know your genes, we'd say, I really wouldn't take this drug, or this is perfect for you. And we can do that in an environmental dependent way, or a developmental stage dependent way. We can do that in an agricultural context across a heterogeneous farm. Like this is the best wheat variety you could grow in the clay soils. But I've got sandy soils over here. All right, I'm going to plant a different variety. That's totally reasonable, but it's really not widespread now. We just kind of put one seed packet and plant it everywhere. But the precision agriculture is optimizing the seeds for even the local environment. And we'd like to include not just these adapted or predicted lines, but we can make sure that we bring across background genetic diversity for the unknown, to avoid the monocultures. Over-optimizing or overfitting without balancing the background diversity is a missed opportunity. That's actually the easy part, but people forget. So back to the holy trinity, genotype, phenotype, environment. We're starting with landscape genetics, just as a way to get diversity. And then we'd like to know how it's related to each other and how distantly related they are. This is genotype alone that comes from the environment, but then we can make these predictions, the landscape genomics predictions. Once you get your core populations and you measure their phenotypes, then we can make associations, genome-wide associations, and that can be flipped around for the genomic selection. If you want to make predictions to assist migration and colonization, new genotypes and new environments, we can do it that way. And without genetics, we're sort of stuck with these assumptions, but that's not the environment. When heritability is low and plasticity is high, just diet and exercise, okay? That's the most important. The environment predicts the phenotype. Let's not get carried away, you geneticists, you bioinformaticians. You might be wrong. Sometimes good old diet and exercise is the cure. So we need to fit all of these components. And the application that I just send you off with here, actually I'll take a little break, save this piece, and I'll come right back.